So I want to welcome everybody back from the break. The first speaker in this session is Josefin Stiller from the University of Copenhagen, and she's going to talk about 10,000 birds.
Hello, everyone. I hope you're refreshed. I want to thank the organizers of this meeting for giving me the opportunity to introduce you to the 10,000 Bird Genome Project, short B10K. And I would like to update you on our progress, on the history, and the goals of this project. The B10K is a collaborative effort to generate draft genomes for all 10,500 bird species to address fundamental questions in evolution, ecology, and biodiversity. If we had a full phylogenetic tree of all bird species, it could look like this. Unfortunately, it's a copy and pasted synthetic tree, and it's not the real deal. But hopefully, in a few years down, we will have this complete tree in order to allow us to address important questions in biology. The B10K is organized in 4 phases, which sample the diversity of birds at varying levels of taxonomic diversity. The goals of the project are to generate a global phylogeny of birds from genome scale data. And this is the ultimate goal for the phylogenomics branch of the project, which I'm coordinating. Our paleontology work group uses the ample fossil record for birds and to integrate it to a time-calibrated tree of bird diversity. And resolving the phylogeny of birds has been a matter of controversy for decades, and it has proven to be actually one of the hardest problems in phylogenetics. And we hope that with genome-scale data and increasing taxonomic sampling, we can achieve an improved resolution of this tree of life. The second major goal is to conduct comparative genomic studies on these bird genomes to investigate the genomic basis of the fascinating bird traits and life histories, and evaluate how environmental and also anthropogenic factors influence bird genomes. Third, the project will generate large public resources that will hopefully stimulate research in a variety of disciplines. These resources are not only the genomic data that we're supplying, such as assemblies, annotations, gene trees, and alignments, but also include expert-curated trait databases for all species that we sequence. The B10K is organized in 4 phases, where the first phase sequenced representatives of all of the major orders. The second phase that we're currently in sampled all families of birds. The third phase aims at generating at least 1 representative for each genus, and then the final phase is to have genome sequences for all of these. The B10K builds on the success of the avian phylogenomics project, which concluded in 2014. The first case was conducted by a large international consortium led by Goji Zhang from BGI and the University of Copenhagen, Tom Gilbert at the Natural History Museum in Copenhagen, and Eric Jarvis back then at Duke University and now at Rockefeller. They sequenced and assembled genomes representing 34 orders of birds. And this project was kind of the proof of concept of the kind of analyses that can be done with this type of genomic data. It resulted in over 50 publications on the phylogeny, comparative genomics of birds, and of which 8 were published in a special issue of Science. But I would like to spend a couple of minutes just justifying why one would want to sequence all species of birds, and just convey some of the fascination about these. Birds are the only living surviving dinosaurs, which makes them cool in their own respect. But the Avian Phylogenomics Project has shown that birds underwent this rapid Big Bang diversification after the Cretaceous-Paleogene boundary about 66 million years ago. This rapid radiation is also the source of very short branches and high levels of incomplete lineage sorting, which make the estimation of the phylogenetic tree challenging. And much of the immense diversity of morphologies, ecological niches, and physiologically adaptations go back to this rapid radiation of birds. Birds enjoy popularity both from scientists, but also from a large number of hobby ornithologists. And for that reason, birds are a pretty good system where we have a lot of biological information of high quality already available. There's a nearly complete global inventory of bird species, which is of course important if you want to sequence every living species. And there are good resources on distribution and biological data. Birds are important to humans. We consume them for their meat, their eggs, their feathers. And chicken and zebrafinch are important model organisms in biomedicine and in behavioral biology. Birds also impact human lives. We domesticated them as pets, but they're also pests in agriculture, and water birds are known to be important carriers of diseases. Groundbreaking ideas were developed studying birds, from Nico Tinbergen's studies on the behavior of animals over Ernst Mayer's theory of speciation, and of course Charles Darwin's studies on Galapagos finches and on domesticated pigeons. Importantly, from a practical point of view of being a genomic consortium, birds are a really good group to study because they have relatively small genomes of about 1 gigabase size, and the Avian Phylogenomics Project has shown that the genome structure is highly conserved among all birds. This simplifies our sequencing efforts, annotations, and assemblies. So here on this synthetic tree, I highlighted in blue the species that were sequenced for the Avian Phylogenomics Project. And I'm now showing where we are currently in our second phase of the project. So we have sequenced and assembled and annotated samples from 363 species of birds, representing at least each family of the group. And we've expanded the board of principal investigators to include experts in museum science and ornithology and biogeography and ecology. Carsten Rabeck from the Natural History Museum of Denmark is in charge of producing the trait databases for all of our samples. Gary Graves from the Smithsonian Institution and Fu Minlei from the Chinese Academy of Sciences are instrumental in supplying tissue samples for our work. It is also important that there was dedicated personnel added to organize and coordinate the efforts of both the phylogenomics branch and the comparative genomics branch. And this is how I came to the University of Copenhagen, coordinating the phylogenomic analyses. And it should be stressed that with a consortium of this size, this is hardly something that one principal investigator can do on the side. Our consortium has grown to a staggering number of over 120 consortium members from all over the world, and they're all really smart and nice. Several funding sources in Denmark and in China have supported phase 2, including the Carlsberg Foundation. Lundbeckfonden has supplied some money for computational resources, and the Chinese Academy of Sciences, and particularly BGI, are important in supporting and conducting the sequencing itself. But I should also mention that there are significant contributions of our consortium members in what the amount of time, money, and resources they put into this project. So a lot of the computational analyses are being done by different groups, and they give us their computational resources as part of being part of the consortium. So this really has to be acknowledged as a significant source. For B10K genomes, we generally require that the tissue comes from a vouchered specimen. So there has to be a physical specimen if anything ever comes up in terms of the identification of the species. or morphological investigations have to take place, we can go back to the museum and look at the animal. Ideally, we want to sequence females, so we can sequence both sex chromosomes, as that's the case in birds. And we have this website, which has a lot of great information, but also information on all of the 10,000 bird species and at which stage we are at, including for sequenced samples, we have annotation reports and assembly reports and reports on where the specimen was sourced from. We have closed the data acquisition for the phase 2 of the B10K project around fall of last year. And here I'm showing the different sources of the 363 genomes of the phase 2. 36 genomes were publicly available, in addition to 42 genomes from the APP project. And the bulk of the samples were generated de novo for phase 2, so 236 genomes. But what I also want to highlight on the far right is that we placed an open call on the evolutionary listserv Evoldir, asking for any unpublished bird genomes of a certain standard. And we received 49 genomes, some of which have the highest, the best assemblies of all of our samples. And this was really quite surprising how supportive the community is to donate even their unpublished data. Some of these genomes have been published in the meantime, but some of them are still not. The different data sources also come with heterogeneity of sequencing technologies and the resulting assemblies. So with our genomes, we are far from EBP standards and from reference gene standards, but I hope I can make the case that we can still generate enough data to answer the questions we're interested in. So here I'm plotting the scaffold N50 for all samples and broken up by the source of the samples. So on the left, you have the NCBI genomes, including some of the model organisms like chicken and the zebra finch. And then in light blue are these samples we got from the community. And they were sequenced with a variety of technologies, from short read technology to long read technology. And you can see that some of the genomes have really good scaffold N50s. For phase 1 of the B10K, all B10K genomes were produced with short read technology. In phase 2, there were 2 different strategies that were followed, and this is kind of why you get this bimodal distribution of assembly qualities. And all of the new genomes of phase 2 were generated with 2 libraries of 500 base pairs and 2 KB size and sequenced to an average of 60x coverage. So our scaffold N50 is below 1 megabase, but nonetheless, when we annotate coding regions across these genomes, we can still annotate a large number of orthologs across all of the genomes. And this is one data type that we're now using for phylogenomics and also for comparative genomics, obviously. The future plans moving forward. Initially, we announced that sequencing of all 10,000 birds would be concluding in 2020, and the sequencing may actually conclude, but probably publications coming out of these will be later. But we are planning the start of phase 3. We have acquired about half of the needed tissue samples from collections, and extractions will start soon. We're also shifting our sequencing strategy with new 10x libraries, and with support from BGI, we can generate a genome at a rate of $1,500 with this technology. And with this, I want to thank you, and I'm happy to take any questions.
One question? One question? No? Okay. Yes, Emma.
I think it's extraordinary that you got the community to give you those 49 genomes. I mean, I do, from getting involved with some of the bird people, they are a lot more friendly than the mammologist side. But was there any, did you have to have any, I suppose, disclaimer with this, or was the rules of engagement a little different? What did you do?
So we basically said that the genomes remain theirs. They were invited to join the consortium. They are invited to use all of the data that we produced. We supplied them with our annotations and with SNP callings, so they're getting a little bit back. But basically, there was no embargo. It's their data. I also have to say that this was closed in November, and since then, there's basically every week people write to us, we have genomes, do you want them? So if we could continuously update this, we would have way more.
Okay, thank you.